Hello everyone, welcome back to my channel. This is your host Jude Stephen. Today's episode is a continuation of the previous episode of drug discovery through regulatory lens. If you missed it, please go back to the previous episode and watch how regulatory oversees drug discovery. Let's dive back to the topic. Cut to 2006, and in London, 6 volunteers were given a new immune therapy called TGN-1412. Everything looked safe. They were dosed with 500 times lesser than the dose that was given to the animals. However, after 90 minutes, they started having multiple organ failures. They were admitted in intensive care and few lost some digits as well. What really happened there? Is it because the animal testing went wrong? Not really. What happened, what worked with monkeys, it just didn't work with humans. This made me think a lot during my compilation of a clinical trial application to FDA. We'll look at that clinical trial application, which is called as IND, in the next episode. Let's come back to the topic. I was compiling this dossier and I had this question: how do you decide what is the safest dose to give to the human as a first dose? And how do we derive it? This led me to a conversation with the non-clinical trial expert, and it was a eye eye-opening topic, I would say. So, how do we do it? In non-clinical, when I'm compiling the non-clinical dossier, we started presenting all the tests that we have done in the labs using cells, using animals, and what all the things that they faced, like the adverse events and all kinds of studies that they conducted. But one thing was not clear is how these animal testing gave the human testing a value addition, or how animal test gave me the first dosing amount or a range on the human beings. It should make sure that there is there is a good amount to show the the effect, but also it should not hurt anyone. That's when the non-clinical expert said we use two safety factors or two safety anchors. The first one is no observed adverse effect level. We call it NOAEL. This is the highest dose in animal at which you see no harmful effect. After this, you start observing animal toxicity and death. Next is MABEL, which is minimum anticipated biological effect level. That gives you, from there, the effect starts happening. Before that, there is zero effect. So in between these two safety anchors, there lies something called therapeutic window. The therapeutic window is what we are looking at. So once you get these two safety factors, you need to convert them to a human range. So it is multiplied through the body surface area or various other factors to get the human equivalent dose. Once you have the human equivalent dose, the guideline S1C, which is a safety guidelines, says that you need to divide it by the safety factor. With regards to the human, it is divided at least 10 times lower. This gives the maximum starting dosing range. Now, after this is what we enter into humans, and this dosing range is mentioned in the clinical protocol. For a small molecule, NOAEL is more than enough. However, for a larger molecule like monoclonal antibody, it's better to have more conservative approach of having two safety anchors. You can never be sure what is safe in animals be safe in human beings also. But that's not what changed my mind about how regulatory sees this. The major thing that changed my mind is the cost. By now, you must have understood how much cost would have gone in. However, let's reiterate. First, the science make you earn the way up. Next, from the computer model, you move to the cell panels. You test them in various cell panels, understand how the drug interacts with the cells, what are the receptors it binds, and that is called as in vitro testing. Once in vitro testing is done, you move it to animals, which is in vivo testing. In vivo testing, it's not directly monkeys. You start from rodents. It could be mouse models. It could be your hamsters, guinea pigs. Then you make yourself move to chimpanzees. It could be brutal in many places and also sometimes called cruel. However, you need to understand how important it is. In our case, testing 1,000 animals costed more tens and lakhs, and one single chimpanzee model or a monkey model costed almost 80 lakh rupees. And according to the guidelines, you must test both on male and female, at least 4 and 4, and keep few of them for a longer range of studies. You do the calculation on and try to understand how much money is involved in here. Next, it's a life. You don't want to waste it. That's why you cannot skip it. Now you can understand, even though it is cruel, why it is important. If you think back about the 6 people in London, this testing becomes more and more critical. For a skip that work, guess the window wrong, and you don't get a smaller trial. You get a cytokine storm, ICU, and the molecule now that's dead anyways. The careful, costly, uncomfortable non-clinical work is what protects the things at once. The human who takes the first dose and the years and crores riding on that molecule. Let's step back and look at what just happened in one single decision of finding the first dosage. Deriving the safety anchors like NOAEL and MABEL and scaling the dose, it's purely science. And weighing the 80 lakh rupees on monkeys and other animal models is the cost to the budget. So that's business. Knowing which guidelines to apply and what all is required for a larger molecules or a small molecule, and that's regulatory. In the non-clinical stage, these three aren't neighbors. They are welded together. And that's the real reason a good regulatory person has to understand all three at once. You can't set a first dose if you only know one of them. There are clever strategies used in this phase too. Things like pooled clone studies, disease model testing for fast track approval, which is used in case of COVID, and high dose approaches to squeeze more insights from a fewer studies. Tell me in the comments if you want a full episode breaking down these strategies, and I'll make it happen. Next time, we hit a major milestone in the regulatory journey. The moment of this non-clinical work has been building towards. If this gave you some idea on how regulatory is involved in the animal studies, please like and subscribe to the channel and share it with someone heading to this field. This is your host, Jude Stephen, and I'll see you in the next one.